Welcome to Module 2.1. This is fertilization. Not really to get too much into the details of the fertilization, but just to understand that this is actually where this process is what starts that genetic recombination. So understanding the male and the female side and where each one of those contribute to uh, like the offspring or the F1 generation. Uh, that's going to give us an idea about how we can go about improving our crops. So embryogenesis is really just the combination of a haploid male, which is one N. And so when we talk about ploidy, typically you'll see this as an N. And then, there might be several types of ploidy, and we'll discuss those in another module. So we have a haploid male with a haploid female, and that produces a diploid or 2N zygote. And so the male gamete is the pollen grain. And so that pollen grain comes from the anther. So on that anther, the pollen is developing in order to fertilize an egg. And so the female gamete is the ovule, and that ovule is inside of the pistil. So, when we go back to the parts of the flower, we've got the stamen and the carpel or the pistil, depending on, some people call it something different. But we know that we have the stigma, the style, and then ultimately the ovary. And so eventually the pollen lands on the stigma, makes its way into the ovary, fertilizes the egg. That's the long and the short of that. So there are two initial processes. So we have microsporogenesis, and that is going to be how the pollen forms as a microspore. And then we have megasporogenesis, and this is how the ovule forms. Okay, so taking a look at this from kind of start to finish, we have the anther with the pollen that lands on the stigma. Ultimately, this is going to grow down the pollination tube and enter into the ovule. The egg and the sperm will unite, form a zygote, and then that will then form the fruit, or well, at least the fruit and then the seed. So, the seed is going to carry on the genetic information for that next generation. So having an understanding of how this is all going to happen, we can start to manipulate if we know the different genotypes of not only the plants, but also the parts of the plants. If we know that genome, then we can, start to really start specifically and intentionally crossing specific pollen with specific ovules, so that we can get the desired result that we want. Like that is probably, I guess that's kind of like going back to Gregor Mendel, as far as he knew that he had a purple plant and a white plant. When he crossed them, they had a certain outcome or a certain phenotype. And if he knew the genotype, then he could predict or kind of dictate what the phenotype was. So that's really all this is just about, is just mostly about this fertilization part, because remember the fruit or even sometimes the seed are what we eat out of these plants or what we use for our nutrition. All right, so just a couple of things to talk about with microsporogenesis. So we have this pollen grain, and this is really just the protective coating. So each one of these are just layers that kind of protect the inside. When I look at this, I think about like a jawbreaker, right? There are certain coatings, the jawbreaker, there's like that very tough outside coating that's really difficult to get into, right? So typically we have to dissolve that. And on the inside, when you get to the center of that jawbreaker, it's a little more soft, and you can dissolve that a little bit quicker. Same concept with our pollen grain, right? And so inside of the nucleus, so after we have the exine, which is that outer coat, and then the intine, which is going to be that kind of inner coat. Uh, that is actually going to end up forming the tube nuclei and the generative nuclei. So inside of the enzyme, that is actually what encapsulates the tube and the generative nuclei. And these two, when I think about this, it really kind of like the tube nuclei. The tube nuclei is basically going to run block for the generative nuclei. Like it's going to be kind of in front, kind of leading the way. If it runs into any problems, that generative nucleus is still going to be safe in the pollen tube. And you'll see this in a moment when we start getting into the whole process. All right, so that was the microsporogenesis, which is going to be the pollen. Next, we are talking about the megaspore or the mother cell. And there are several divisions. Again, I did not want to make this a genetics class or start getting into a whole bunch of microscopic cellular concepts. But there are a few things to understand that this megaspore cell ultimately turns into this embryo sac. And within those embryo sac, there are certain components that are important in order for this fertilization to not only the fertilization, but also like the continuation of that. It's one thing to just merge a sperm cell with an egg cell, but there's some other components of that that need to be carried on with that so that that embryo can survive. And then also when it's time for it to germinate and grow, it still needs some things like food. So ultimately, the two polar nuclei end up becoming the endosperm, and that endosperm is what's going to give that seed its food when it germinates. That's kind of where I'm heading here. This is kind of the purpose of understanding this is like, this is the genetic information and how it gets to actually making that seed. And then when that seed germinates, we see the phenotype. All right, so just to walk through that, we've got this diploid cell that separates into 4 haploids. We have a few degenerated, a few cells don't make it. One of them does, and that continues on through this process. Again, I'm not really concerned about mitosis or meiosis or any of those kind of biology concepts. I think, as an upper division class, we understand that there are a couple of divisions that happen such that we get that crossing over and we get that genetic recombination. I think that's probably that in a nutshell. We're going to have our synergids and our egg. We have the polar nuclei, and then we have our 3 antipodal cells. All right, so inside the ovule, there are a couple of components. And what's important to understand about why these components are important is because they end up becoming something or some part of a fruit that we eat. And so each time that we have a different fruit, the integuments may be this part, or inside of a seed, we might recognize that is where these particular components of that seed are. And that's why it's important to at least be aware of what we are trying to change. All right, so we have our integuments, and the integuments really just kind of protect this nucellus along with this megasporocyte. Remember, this is going to end up becoming this. It's going to, this is our megasporogenesis, right? And so then we have the ovary wall, and these actually become kind of the parts of the fruit that we eat. So, you can even look at this and kind of go, that kind of looks like a peach, right? So we have, kind of our fleshy area. We have our rind or kind of outer area. And then inside of it, we have like that pith or that stone. Next, we have the micropyle. And the micropyle is just, it's kind of like that one way in, one way out. So there aren't just a lot of opportunities in order for that sperm to come in. It has to come in at a specific spot to make sure that it is fertilizing only the egg. Okay, and then finally we have the embryo sac. And within that embryo sac, those 8 cells from megasporogenesis, these 8 cells are located inside of that. And we have 3 cells here. One is the egg cell and our 2 synergids. And our 2 synergids are kind of like the bookends that kind of keep that egg in place and also kind of give it some protection so that it's in the proper position so that it can be fertilized by that pollen grain. Next on the chalazal end, not that you'll ever use that again. I wouldn't think unless you intentionally go into plant breeding. But on the other end of the micropyle, there are the antipodals. And then finally in the middle, we have our 2 polar nuclei, and these also play a role down the road when we're thinking about double fertilization. All right, so going through the whole process of germination. So we have our pollen grain that lands on the stigma, and then this will start to grow down the style into the ovary. So we have this pollen tube, my mouse, the pollen tube that will take one of these grains down into this embryo sac where the egg and the polar nuclei are, right? So one thing that's important about this pollen grain, there are certain signals inside of, certain chemicals and certain reactions that act as indicators so that the plant knows if it's self-fertilized or if it's cross-fertilized, or I'm sorry, self-pollinated or cross-pollinated. So if it's self-pollinated and let's say another, the pollen from another plant gets on that stigma, my mouse. If pollen grain from another plant falls onto the stigma, then nothing will happen because the stigma, there are these receptors in there that tell if it's at least of the same plant species, and then also if it is of the same plant or a different plant. So for this one, it's really just that it's the same plant species. So as that pollen grain reaches that stigma, those absorbed nutrients are going to develop the pollen tube, and that will then kind of act as the channel or the tunnel that gets that pollen grain to the embryo sac. If you remember, I talked about the tube nucleus. That is actually going to be at the growing tip and kind of leading the way and making sure that there's nothing that's going to stop or impede that tube nuclei, I'm sorry, the generative nucleus from getting to the egg. So again, it's kind of running block. Once that pollen grain or the generative nucleus gets to the gametes, once the generative nucleus gets into the embryo sac, it will divide into 2 gametes or nuclei, which kind of move to the tip of the pollen tube. Each one will do the same thing. So the tube nucleus just runs block. It will dissolve by the time that it gets to the embryo sac. Once its job is done, it's done. Next, the generative nucleus will divide into 2. One will fertilize the egg. The other will then move to the polar nuclei. All right, so just to look at this again, a little better picture, we have our pollen grain landing and growing down the pollen tube, the generative and the tube cell, remember? So here we have our tube cell that's running block for this generative cell. As we get to the end, this tube cell is going to disintegrate, and the generative nuclei will divide into 2 haploid, that's 1N, 2 haploid sperm cells. All right. Once that makes it there, those 2 sperm cells are released from the pollen tube. One is going to fertilize that egg. The other is going to unite with the polar nuclei. And that is actually going to form the endosperm. All right, so once that happens, there are two sets of fertilization. That's why it's called double fertilization. So we have the sperm nucleus and the egg cell, which is what we want, what we want, or we have the sperm cell and the definitive nucleus. And so if one, then the other fails, right? So if the sperm nucleus fertilizes the egg cell, well, then we get what we expect is the normal fertilization. If the egg cell fails to fertilize, that other synergid will then assume the role of the egg cell, and then it will be fertilized. And so once that egg cell is fertilized, that's where meiosis takes place. And that's really where that genetic recombination. And so, kind of the purpose of this YouTube video, if you haven't watched it, go back, it'll kind of tell you the same thing that I've done it probably in a better voice. And, it's pretty straightforward. It's got some demonstrations of like what actually happens. The most important thing to understand about that YouTube video is that is where the alleles and the chromosomes and everything starts to line up, and you can see that genetic recombination, and you can see that it's really kind of a chance thing. It's kind of predictive, but kind of not. We don't know which one that is going. We don't know which recombination we're going to get. It's sort of random, kind of. So the goal is that if we can make sure that the sperm cells and all of the alleles are big B in both the male and the female, then we only get big B offspring. Is that making sense? So by being able to manipulate the male side of the flower and the pollen, and making sure that the genotype or that the alleles that are going to end up fertilizing with that egg have big B, or that they are homozygous dominant, that is going to help us create lines, and that is going to help us kind of predict and ensure that we get the right offspring that are going to benefit us down the future. And that's kind of what we'll talk about in the next modules, is like, how do we go about developing a line? How do we go about, the old traditional methods of plant breeding in order to kind of develop these characteristics? And then like the second half of the next module is going to be like, we really don't have the time for all that. Here are some other techniques that we can use in order to kind of speed that process up, because we need that food now. And we need to have these different phenotypes and genotypes ready to go and to be put in our fields to be grown to feed ourselves. So kind of to sum this up, like what's the whole purpose of this and why are we going through this little part of it? Well, the main thing is that genetic recombination. And once we know where that genetic recombination is occurring, once we can dictate what does or doesn't occur in that genetic recombination, then we can start developing these lines and different hybrids that are going to have that hybrid vigor, that are going to have those resistance, pesticide, insecticide, herbicide resistance, disease resistance, etc. If we know how to change that genetic recombination. Also, in the next module, we're going to start talking about the difference in self and cross pollination and how that helps us develop those hybrids and those lines. So, knowing the genetic recombination, we can start to predict the potential outcomes of those crosses and whether they're self or cross pollinated. If we can manipulate both sexes of that plant, then we can start to streamline the phenotype or the phenotypic outcome that we want for that. And so this is really what this is leading down to, is like, how do we go about developing these lines? And ultimately what we want to do is maintain that genetic consistency, even though we have increasing populations. And also not only maintaining the genetic consistency, but also kind of a segue from module 1, we also want to maintain some of that genetic diversity, so that it's possible that we could end up taking these seed that have these traits that might end up being crossed with a different seed that have different traits, and make a completely new population of plants, should that ever occur. So, when we develop these hybrids in these lines, when I'm thinking like Pioneer, or I'm thinking DeKalb, Asgrow, when they're developing these hybrids in these lines, that genetic information is being stored in one of those repositories, so that just in case if they ever needed to come back to those genetics, that they have them. So this isn't just like crossing a bunch of plants and just hoping and praying that it works out. Like, this is an intentional decision and an intentional process, and we're going to keep a hold of that because we don't want to lose that. They've done all the work for it. So it's not only maintaining that genetic consistency, but it's also going to preserve some of that genetic diversity, because we have now kind of genetically modified that plant. All right, so that's all there is for this module. I just wanted to kind of go through that process just a little bit, and kind of show you where we're headed in the next modules, to get toward this self and cross pollination. And when we start doing this, you'll understand and go, wow, that's a lot of work. Why would we do that when we can get 8 years worth of work in 2 years worth of genetically modified or genetic engineering in order to get to the result that we want to faster? Okay, that's going to be it for module 2.1.